Hello everyone. This week in lab we're going to talk about biochemical tests. Those are tests done to help us identify the bacteria that we are looking at. So we are going to specifically talk about biochemical tests that would help us identify different enteric bacteria. Enteric bacteria, those are the ones that you can find in your guts. Whether those enteric bacteria are part of your normal flora or whether they are pathogenic enteric bacteria. Those biochemical tests that we are going to discuss are going to actually help us identify those various enteric bacteria. And those biochemical tests, they are pretty important because you cannot just rely on, let's say, staining techniques to be able to identify the bacteria. So if you have a patient and this patient, is presented to you with a bunch of different symptoms, you cannot just based on the symptoms exactly know for sure what type of infection do they have. So we usually take a sample from our patient, whether that's a urine sample, a fecal sample, or a nasal swab, a sputum sample. So you go ahead and take a sample from your patient, and then you go ahead and you start doing some tests. Now, of course, you can do, as I said, you can go ahead and stain the sample, and you can do a gram stain, an acid-fast stain, an endospore stain, which is definitely going to give you a whole lot of information. But then on top of that, you need to do some biochemical tests. And so we're going to talk about these various biochemical tests, how to read them, how to interpret them. So let's go ahead and get started. Biochemical tests, as I said, they're going to help us identify the microorganism. Now, based on what? Based on two things that we are mostly going to discuss today, based on what they can metabolize in terms of nutrients, what type of nutrients they can metabolize. For example, can this bacteria metabolize proteins? Can it metabolize carbohydrates? Now, if it's capable of metabolizing carbohydrates, what type of carbohydrate it can metabolize? Can it metabolize glucose? Can it metabolize lactose, sucrose, or maybe all three of them? Okay, so it's going to help us figure out the type of bacteria based on what type of nutrients they can metabolize. Another thing is we can identify the bacteria based on the enzymes that they make. Now, not all bacteria out there make the same enzymes, and so therefore we can use that to help us identify the different bacteria. Now, we usually, to identify the bacteria, after doing a bunch of biochemical tests, and we get the result of each biochemical test, we go ahead and we take a look at something called a dichotomy scheme. It's kind of like a chart, and you go ahead, take the results that you just gotten after, performing all of the biochemical tests, use this chart to help you identify the bacteria. I'm going to show you a dichotomy scheme. We're going to talk about exactly how to use it and how to read it. Now, for this PowerPoint, we're going to focus on enteric bacteria. As I said, those are the bacteria that live within our guts. Those could be part of our normal flora. And then, of course, there are some that are pathogenic. So those are the ones that could make us sick. Now, enteric bacteria, they fall under a family called Enterobacteriaceae. Now, what's a family? Remember when we were talking about taxonomy? And so you have that taxonomy hierarchy, the domain, the kingdom, phylum, okay, class, order, family, genus, species. And so Enterobacteriaceae is basically the category the family category, okay? Now, there are a few features, okay, about the Enterobacteriaceae bacteria that you should know, and that is Enterobacteriaceae bacteria. They are gram-negative, so when you go ahead and do the gram stain, they're going to turn out pink under the microscope, so they are gram-negative, and they are bacilli. They are rods in shape. Another thing about them that you should know is that they are oxidase negative. It means that they do not make an enzyme called oxidase. And oxidase is an enzyme that's used in the electron transport chain. And the electron transport chain is mostly associated with aerobic respiration when there is oxygen. So these bacteria are oxidase negative. They don't make the oxidase enzyme. And so what does that tell us? It tells us that this bacteria can actually grow without oxygen. Okay, and if you think about it, as we said, those are bacteria that live within your guts. So it makes sense that they can live without oxygen since they're going to be within our guts. Okay, so where there isn't oxygen, and so therefore it makes sense that they would be oxidase negative. Another important feature about them is they can ferment glucose. Okay, so they have the ability of fermenting glucose. Okay, and we'll talk about the test that can help us determine whether they can ferment glucose or not. But you know, they do ferment glucose, and so these are three important features about Enterobacteriaceae bacteria. Okay, they're gram-negative, they're bacilli, they're oxidase negative, and they are glucose fermenters. Now, let's go ahead and start talking about those biochemical tests. So, one of the tests that we're going to talk about is a broth test. So, it's done in a broth type of media. Remember how we said we have different types of media? We have broths, we have deeps, we have slants, we have plates. So these biochemical tests, by the way, they can be done in all of those different types of media. So we're going to start with a test that's usually done in a broth type of medium. Okay, so that's why we call it a broth test. But what's the specific name for it? Well, this test that we're going to talk about right here is called the carbohydrate fermentation test. And this broth that you are seeing right here, this is an image of it that's when it's uninoculated, meaning that you didn't introduce any bacteria into this broth yet. Why is it important to actually see an image of you know, this carbohydrate fermentation test when it's not inoculated? Because you want to know color-wise, okay, want to know how does it look like, what is its color before I even introduce any bacteria in it, so that it can kind of help you figure out what's happening after you've inoculated it, because what's going to happen is the color is going to change, okay? And this is how we can tell. But before I go over the detail, let me again talk about the name. So the name of this test that we are specifically talking about, it's called the carbohydrate fermentation test. Or sometimes they refer to it as the Durham test, okay? So the Durham test or the carbohydrate fermentation test, or sometimes they even call it the sugar fermentation test, because carbohydrates, they're sugars, right? So sometimes they refer to it as the sugar fermentation test. Now, what's the Durham, by the way? A Durham is a tiny, it's kind of like a small, narrow tube that's placed in that large tube. So here's the large tube, right, that's filled with the medium. And this medium usually comes in that color. It's kind of like a peachy color, okay? And again, it's that peachy color because it's uninoculated, okay? And by the way, it's a peachy color because we add to it an indicator, a pH indicator. And when that pH indicator is neutral, it's kind of that peachy color that you see right there. And then inside, there is this narrow tube. It's an inverted tube, okay? So you can kind of see how right here, the tube is actually closed up because it's inverted. And we're going to talk about what's the point of adding that Durham tube inside here. Okay, there's a purpose for it, so we'll talk about it. Again, this is how it would look like uninoculated. Now, how should I inoculate, this carbohydrate fermentation tube that I have right here? Well, since it's a broth, right, we're going to go ahead and use a loop. Okay, so you're going to use a loop, and of course you have to, so if here's your loop right here, you have to follow all the aseptic, right, steps that we've talked about earlier. So you would have to sterilize your loop, until it turns red, kind of wait a second, and then go ahead and pick up some bacteria on your loop, right? You don't want to grab a whole lot of bacteria. So on the tip of your loop, you would want to pick up a sample, and then you would go in here, okay, and you would basically, stab, or not stab, I mean, it's a broth, right? You would just kind of go in, okay, and go ahead and deposit the bacteria that you have on the tip of your loop, okay? So after you kind of inoculate, of course, your broth, right? You would have to go ahead and incubate it. So you would go ahead and incubate it. And then after 48 hours, you would go ahead and start looking at your results. Now again, what's the whole purpose of this tube? Well, the purpose of this test is we want to know whether your bacteria can go ahead and ferment carbohydrates, whether it has the ability to ferment sugars, okay? And usually in this broth, we usually add only one type of sugar. So this broth in front of you, let's say, only has glucose in it. And then I might make another one that only has lactose in it, etc. So you would only add one sugar in this broth, and you're trying to figure out whether your bacteria can ferment that sugar or not. So let's take a look at some of the test results that you would get, okay, after incubating your carbohydrate fermentation tubes. So one more time before I show you the results, it's going to test for the fermentation, okay, of one carbohydrate, okay, guys. So usually, again, these tubes would only contain one type of sugar. Let's say glucose, okay, so it's only going to contain glucose, or it's going to contain lactose. Again, one sugar at a time. We usually do not add more than one sugar, okay, in this test. We only add one sugar, okay? And this test then will help you determine whether your bacteria can ferment glucose or whether it can ferment lactose. So, and as I said, how are we going to figure it out? Well, based on the color change that you're going to see, okay? And why would a color change happen in the first place? Well, because these tubes, as I mentioned earlier, We add to the broth, okay, a chemical, and this chemical is a pH indicator. What's an example of a pH indicator that we use with this test? It's a pH indicator called phenol red. Now, phenol red is a pH indicator. What does that mean? It means that based on your broth, based on whether your broth is acidic, basic, or neutral, that phenol red is going to change color. And so based on what color we're going to see, we can tell whether our broth is now acidic, basic, or neutral. Okay. Now, if the phenol red turns yellow in color, that would tell us that the broth is now acidic. If the phenol red turns pink, that would tell us that it's basic. Now, if it's orange, that would tell us that it's neutral, okay? And so remember that image of the uninoculated tube that I showed you? It's that peachy, peachy orangish color. That's actually neutral. So we usually start with a neutral uninoculated tube, and then based on if it stays, orange, or if it turns yellow, or if it turns pink, we can go ahead and determine what's happening. Now, the thing is that if the bacteria goes through a fermentation process, when a bacteria ferments carbohydrates, one of the main products that will result is acid. So we know that if the bacteria is capable of fermenting a sugar, then the solution is going to turn acidic. Okay, so if it's going to turn acidic, then the color of the test tube is going to turn yellow. Okay, now if your carbohydrate fermentation broth doesn't turn yellow, if it stays orange, or let's say it's pink, then that would tell us, again, pink is basic, so that would tell us, well, your bacteria is not capable of fermenting the carbohydrate. Okay, so let's go ahead and kind of take a look at some of the results together to make more sense of what I'm saying. So let me kind of first list some of the colors that you might see, and see together what would that mean, and then I'll show you the images. So if your broth is red or kind of orangish in color, well, remember how we said we kind of start off with that peachy orangish color? So that could just mean, well, this is just an uninoculated test tube that you have, right? So it's not inoculated, okay? And if it's, of course, if it's not, let's say, well, you're not sure whether this tube is inoculated or not, okay? How can you tell whether it's uninoculated or whether it is inoculated? Well, look at the turbidity, right? If there is no turbidity, right, if it's lacking turbidity, then of course it's, it's not inoculated, okay? Now, if the broth is yellow, that would mean it's positive. That means that your bacteria is positive. Positive for what? Well, it's positive for carbohydrate fermentation. It means that my bacteria is capable of fermenting sugar. Now, either it's capable of fermenting the glucose or it's capable of fermenting the lactose, depending on which sugar you added in your broth in the first place, okay? And that would mean that now the broth is acidic, because as I said, if your bacteria is capable of fermenting a sugar, it's going to release acid. So it's going to turn yellow. Now, if it's pink, okay, if it kind of turns into that pinkish color, then that would mean it's negative. Negative for carbohydrate fermentation, meaning that your bacteria is not capable of fermenting the sugar. Okay, now you might wonder, like, why would bacteria, want to ferment sugar in the first place? Well, to get their nutrients, okay? So you might be wondering, well, okay, if my bacteria cannot ferment carbohydrates, what are they going to be using, right? What are they going to be using for nutrients? Well, the broth usually has other stuff other than carbohydrates. We add in the broth proteins. And so if you see that your broth turned pink, right, it went from that red orangish, color, which is not inoculated, and it turned color, and now it's pink. Well, it tells you that your bacteria grew and everything, but it wasn't able to ferment the carbohydrates. But it was capable of using the other nutrients in the broth, such as the protein. So it was capable of using the protein, but it was not capable of fermenting the carbs. Now, the gas bubble, that's actually, we can determine that using the Durham tube. Remember that narrow inverted Durham tube? Now, this inverted Durham tube, its main purpose, why do we add it? Because some bacteria out there, when they ferment the sugar, they actually release some gas, such as carbon dioxide. Now, and that's just an example, right? Like the gas, it could be carbon dioxide. And so that carbon dioxide is actually going to get trapped in the Durham tube. So if you see gas bubbles in the Durham tube, that would tell you that the bacteria was capable of producing gas. And by the way, if your bacteria is capable of producing gas, then that means it was capable of fermenting the sugar, right? You will never find gas bubbles in a pink broth because remember, if it's pink, it means that it's not capable of fermenting sugar. So if it's not fermenting, right, and if it's not going through the fermentation in the first place, then there won't be gas. Okay, so gas. If you do find gas, then you will find it in the yellow broth. Okay, but can I have a yellow broth and no gas? Yes. Not all bacteria that ferment carbohydrates will produce gas. So one more time, you can find a yellow broth with gas bubbles, or you can find yellow broth without gas bubbles. Okay, but you will never find a pink broth with gas. Okay. So these are, again, some images to kind of help you out. So we start off right here. Okay, this is the red-orange broth. This is the uninoculated. This is the one that doesn't have any bacteria growing in it, right? And then let's say that we go ahead and we use our loop and we introduce, right? We inoculate this broth with some bacteria. And then you incubate it for like 48 hours. And then let's say that you get this result over here, okay? Now you can see a huge change, right? See how we went from that peachy color to that yellow color. Okay, let's interpret the results together. Yellow, that means that it's acidic, okay? And so what does yellow acidic broth means? Well, it means that my bacteria was capable of fermenting the carbohydrates because as I said, fermentation of carbohydrates will result in acid production. So it's going to turn yellow. Now, right here, here's the dram tube, okay? And then do you see this bubble in there? Now, this bubble represents basically gas trapped in the dram tube. So it tells you two things about this bacteria. It tells you that this bacteria that you inoculated right there was capable of fermenting the carbs, and it was also capable of producing gas. Now, what type of carbohydrate was it capable of fermenting? Um, Depending on which carbohydrate you added in the broth. So if this broth had glucose, then I would say my bacteria was capable of fermenting glucose. Let's say this tube right here had lactose, then I would say my bacteria was capable of fermenting lactose. You must be told what type of sugar was added, right? You can't just look at it and say, oh, definitely, there was glucose in there, or definitely by the looks of it there's lactose. You can't, okay? So you must be told what type of sugar was in the broth to start off with, okay? Now, let's say that after you've inoculated, okay, your tube, it kind of turned from that peach color to like that pinkish color, okay? Now, pink means basic, and so that means that your bacteria was not capable, okay, of fermenting the carbs. Why? Because as I said, if it was capable of fermenting the carbs, then acid will be produced. So if it's pink, well, that means that your bacteria did not go through the fermentation process. The bacteria did grow, okay, so you do have growth and everything, and you can also determine that by the turbidity, but because it's pink, that would mean it's negative for carbohydrate fermentation. And so look at the dram tube. Okay, here's the dram tube. Notice how you don't see that gas bubble, because remember, if fermentation did not take place, then you won't have any gas production, okay? So you shouldn't see any gas bubbles trapped in the dram tube. So expect to interpret results like this if you are given a set of images. Expect also to be given an image and identify what test is that, okay? Just by looking at the image. If you see a Durham test inside of a test tube, then you know right away that this is the Durham carbohydrate test. Okay, so again, you need to be able to interpret the results. So know the colors, okay, that you should expect. Know which color is positive, which color is negative, and then again be able to identify the images, okay? So you need to know that just by looking at this image, you need to know that this is the Durham carbohydrate test. We're going to talk about another broth test, and this one is called the Indo Test. And the Indo Test, it's broth, it's called the tryptone broth because it contains tryptone. Tryptone is actually like a protein that has a bunch of basically it's made up of like multiple peptides. And we'll talk about what this test tests for, but this is how its broth looks like uninoculated. Okay, it's kind of like that clear, very faint, like yellowish color. This is another one. Okay, it's like a much darker broth compared to the Indole, and this test is called the MRVP. Again, this is how its broth would look like uninoculated. MR stands for methyl red and VP stands for Vogus Prosker. And we'll go ahead and talk about what those two tests are for. So let's start off with the Indo test, okay? And we said that the Indo test, the broth usually has what's known as tryptone, okay? Now, what's the point of this test? Well, this test is going to tell us whether your bacteria is capable of breaking down a very specific amino acid. And this amino acid is tryptophan. So we want to know whether our bacteria is capable of breaking down the tryptophan amino acid. Now, it's going to tell you whether your bacteria is capable of breaking down that amino acid, and if it does, then the bacteria is going to produce a product called indole. So if tryptophan is broken down, it's going to be broken down into something called indole, and that's why we call it the indole test, okay? Because the indole is going to be the product of the tryptophan being broken down. Now, as I said, we usually start off with a broth that contains tryptone, right? As I said, it's that protein, and proteins are made out of amino acids, right? So it does contain the tryptophan amino acid, and we're trying to figure out whether our bacteria can break it down. If it is capable of breaking it down, then the product indole will be produced, okay? Now, how do we usually do that? So you get your broth, right, that has the tryptophan in it. Okay, you go ahead and you inoculate it. We use an inoculating loop. Since it's a broth, we use the inoculating loop. You go ahead and you inoculate your broth. You put it in the incubator for 48 hours. After 48 hours, you go ahead and you take it out of the incubator, and then you go ahead and add a reagent. Now, not all tests you get to add reagents to it. So, I mean, we just talked about the Durham carbohydrate test, and we didn't add any reagents. This one, you do. But when do you add the reagent? After the incubation, okay? Not before. Okay, after the incubation. So after you've incubated your test tube for like 48 hours, you take it out and you add a reagent called Kovac. Now, you add about 4 drops. Okay, so you add about, this is the reagent, okay, Kovac's reagent. So you go ahead and add directly into your broth, okay, 4 drops of the Kovac reagent, okay? And then you go ahead and start interpreting the results. Now, let me show you first the 2 colors that you would get, and then I'll show you pictures. So if you get right here, yellow band, so again, here is your tryptone broth. As I said, this is after you've incubated your, this is after you've inoculated it and you've incubated it, right? You take it out of the incubator, put 4 drops, okay, of the Kovac, and then notice how right here you get this yellow band up at the top, and the results are usually going to be up at the top, right? So this is where you want to be looking. So you want to be looking up at the top. If you see this yellow band up here, well, this is a negative result. So this tells you that your bacteria cannot break down tryptophan. Okay, your bacteria just can't break down that amino acid tryptophan. But if you go ahead and add the Kovac to this test tube right here, and you get this pink band, then it's positive. This tells you that your bacteria can break down tryptophan. And if it's capable of breaking down tryptophan, then your bacteria has released what's known as indole. Okay, so I would say this test right here is positive for indole. Okay, the bacteria is capable of producing indole. And how do I know it's capable of producing indole? This pink band that you see up at the top. This one is negative for indole and means, again, your bacteria is not capable of breaking down tryptophan. Again, you need to know the colors, okay? You need to know which one is positive, which one is negative. Again, you need to know that just by looking at an image, you need to know that this is an Indo test, okay? Even if it's not labeled or anything for you, just by looking at it, you need to know, okay, this is an Indo test, okay? Now, another test we're going to talk about is the methyl red Vogus Prosker test. We've seen already the uninoculated broth and how it has that yellowish color. So let's talk about, okay, this broth test, what is it for? What is it testing? Well, this test is testing for waste products of glucose fermentation. So you want to know whether your bacteria, when it ferments glucose, whether it's producing an acidic waste or whether it's producing a neutral waste. Okay, um, so we'll go ahead and talk about that in more detail. So usually for you to figure out whether it's producing an acidic waste or a neutral waste, you actually have to have two test tubes, okay? So you gotta have two test tubes, okay? And for one test tube, you're going to add methyl red, and for the other test tube, you're going to add the VP, okay? So one test tube is going to be for the methyl red, and then the other test tube is going to be for the Vogus Prosker. You can't test the two in the same test tube, okay? And again, I'll show you an image to make more sense of what I'm saying. So right here, okay, so you would do a methyl red test, okay? So you would basically, take some of that bacteria culture, okay, and you would put it into an empty test tube, and then you would go ahead and do the methyl red test. Now, to do the methyl red test, you would go ahead and actually add 5 drops of methyl red. And if your broth turns red, that means it's positive. If your broth remains yellow, that means negative. Positive meaning what? Negative meaning what? Well, if you did the methyl red test after adding the 5 drops of methyl red, if it turns red, that means that your bacteria was capable of fermenting glucose, and it's basically producing an acidic waste, okay? Now, remember how I said when bacteria ferments, right? It produces an acidic waste. Now, so the methyl red is going to help you also determine that, right? Whether it's producing an acidic waste or not. Now, the VP test is going to tell you whether your bacteria is capable of fermenting glucose, but it's actually producing a neutral waste. So sometimes, again, some bacteria, when they ferment glucose, they ferment it in a different way, okay? So not all bacteria ferments glucose in exactly the same way, okay? So some bacteria out there, when they ferment glucose, they don't actually produce an acidic waste, even though, again, that's like a common, that's the most common thing, an acidic waste to be produced, right? When bacteria ferments sugar. But some bacteria out there, when they ferment glucose, they release a neutral waste. And so the VP test is going to help us determine that. So for you to do the VP test, okay, you get your bacteria, and then you add to it 10 drops of reagent A, and then you add another reagent. You add 5 drops of that second reagent, and then don't read your results right away. You gotta wait 45 minutes. So wait 45 minutes, and then after 45 minutes, you can start interpreting your results. Now, if your broth turns red, okay, mostly towards the top, that means positive. But positive for what? Positive that your bacteria is producing a neutral waste, okay, when it's fermenting glucose. If it's yellow, that's negative, okay, it's not producing a neutral waste. Now, you can only have one positive test. No way your bacteria can produce an acidic waste and a neutral waste at the same time. It's either or, right? My bacteria is either producing an acidic waste or it's producing a neutral waste. So meaning my test is either going to be only positive for the methyl red test or only positive for the VP test. You will never have positive for both, okay? But can you have the methyl red test to turn out negative and the VP test to turn out negative? Yes, okay. Uh, Again, like, if your bacteria is not even capable of fermenting glucose in the first place, then your methyl red and your VP will be negative, right? So negative for both is absolutely fine for you to get a negative for both tests. But when it's when it's a positive result, only one of the tests, either methyl red or VP. Again, because either it's going to produce an acidic or it's going to produce a neutral waste. So let's take a look at some images, okay? So one more time, the methyl red test is going to test for that acidic waste, right? And that acidic waste is basically the product of glucose fermenting, right? So we know that your bacteria is, if you get a positive methyl red test, right? So if you get a positive methyl red test, it's kind of telling you two things. Well, it's telling you that your bacteria is fermenting glucose and it's producing an acidic waste, okay? And as I said, you get your bacteria culture and you add to it about 5 drops of that methyl red, okay? And then interpret the data, okay? So after adding the 5 drops of methyl red, take a look at the results. Notice how right here this one is yellow, okay? Well, this is negative. This tells you that this bacteria is not producing an acidic waste, okay? Now, if the broth turned kind of that orangey, kind of red color that you are seeing, that means it is positive for methyl red, meaning that your bacteria is producing an acidic waste from fermenting glucose, okay? Now, it's glucose, you guys, okay? Nothing else. So let's say that in this broth, I didn't add glucose, I added lactose. Is my methyl red gonna turn red? Is it going to be positive? No, it has to be glucose, okay? So again, these sugars, don't just think like all sugars are the same. So bacteria are going to, bacteria have the capability of fermenting all the sugars out there, or, when they ferment these sugars, they're going to ferment them in the same exact way. No, okay, that's not the case. So this, again, test is specifically, for the waste product, specifically for glucose fermentation. Okay, so with the methyl red, by the way, when you get the positive result for the methyl red, the whole entire broth turns red. Okay, so when the whole entire broth turns red, that means again that your bacteria is producing that acidic waste from fermenting the glucose. Now, for the VP test, what we're trying to figure out is if your bacteria is producing a neutral waste, okay, from fermenting glucose. So you get your bacteria culture, you add 2 reagents, okay? From the first reagent, you add 10 drops of it, and then you add 5 drops of reagent B, and then make sure that you wait 45 minutes. You gotta wait, okay? Don't just read the results right away. You gotta wait for those 45 minutes to pass and then start interpreting your results. If it stays yellow, again, that just means negative, okay? No neutral waste was produced, so again, it's negative if it's yellow, okay? Now, if it's positive, it's going to turn red. But notice how it turns red like up at the top, like not the whole entire thing turns red. And this is, this is kind of what how it should look like, okay? So it's going to be red up at the top. And so if it's red up at the top for the positive Vogus Prosker, that means that when your bacteria is fermenting glucose, it's producing a neutral waste. As I said, you will only have one positive test, either positive methyl red or positive Vogus Prosker. So actually, guys, like those 2 tests, the Vogus Prosker and the methyl red, we do them together, right? So if you're gonna do a methyl red test, you gotta do a Vogus Prosker test, okay? As I said, you only get one positive, okay? Either it's positive for methyl red or it's positive for Vogus Prosker. You will never have positive test for both. But yes, you can have negative test for the methyl red and at the same time a negative test for the Vogus Prosker, okay? Okay, now we move on to a slant test. So the slant test is, here's like a slant test tube. So here's the slanted part. But this slant looks a bit different because it has a lot of this media down here. So there's this deep part. So actually this part is the deep, and then this would be like the slanted part right here. So when you want to inoculate this test tube, before actually we talk about what is the test called and everything, if you have a slant like this and you want to go ahead and inoculate it, what we do is we inoculate the surface, okay, using a loop. So you inoculate that slant surface using the loop, okay? So using the loop, you go ahead and inoculate the surface. And then to inoculate the deep, we use a needle. Okay, so use the needle and you basically go ahead and you stab it. Okay, so you stab it with the needle. Okay, so we use 2 different tools with this right here. The loop to inoculate that slanted part, and then the needle to inoculate the deep part, okay? So now what is this test called? This is called the triple sugar iron test, or TSI. Now this is how it would look like if it's uninoculated, meaning there is no bacteria in it yet. You would kind of have this, again, kind of like reddish orangish color, and this is called TSI. T stands for triple, S for sugar, and then the I is for the iron. Now, why do we call it triple sugar? Because it has 3 sugars, okay? This kind of, slant right here actually has 3 sugars added. So what are those 3 sugars added? Well, let's go ahead and kind of divide. Okay, so this is kind of like the deep part, and this is the slant. So there's the slant up on the top, okay, and this part is the deep. Okay, now The deep, again, this includes 3 sugars, okay? It has glucose, okay? It has lactose, and it has sucrose, okay? So there are 3 sugars added in here, okay? Glucose, lactose, and sucrose, okay? Okay, these are the 3 sugars that we usually add, okay, to the triple sugar iron test, okay? Glucose, lactose, and sucrose. Now, the top part, the slant, is actually going to be used to figure out whether your bacteria, okay, so this top part right here, we want to figure out whether our bacteria is capable of fermenting. Okay, so whether it's capable of fermenting lactose or sucrose, or both. Okay, so sucrose up here, and again I'll explain it in more detail. And then we look at the bottom portion, okay, to figure out whether your bacteria is capable of fermenting glucose. Okay, so down here, okay, based on the color change, and we'll talk about the color changes. Okay, so you look at the bottom portion to figure out whether your bacteria is capable of fermenting glucose. You look at the upper half, right, that slant part, to figure out whether your bacteria is capable of fermenting lactose and sucrose, or lactose or sucrose. So sometimes the bacteria can actually have the capability of fermenting lactose and sucrose, both of them. And then sometimes the bacteria is capable of fermenting just one, lactose or sucrose. Now the thing is though, you won't be able to find out whether it's fermenting either or both. The results cannot really give you that much information. So I'm going to show you the images and then we'll talk about it more in detail. So remember how this test, it's called the triple sugar, because again, there are 3 sugars in there, the glucose, lactose, and sucrose, okay? But then there's something else, there's iron. And so iron is actually going to be actually used to help you determine a certain production of gas, okay, hydrogen sulfide. So we're going to talk about that in just a minute. So the triple sugar iron test, you guys, tests for 2 things. Tests for carbohydrate fermentation, and it tests for hydrogen sulfide gas production. Okay, so let's go ahead and take a look at the results together. And well, before talking about the results, let me just kind of reiterate. So this test, this slant deep, has the 3 sugars, the glucose, the lactose, and the sucrose. And then there's also iron that's added in there. And we said that the iron is actually going to help you figure out if there is hydrogen gas production. How am I going to know whether there is hydrogen gas production? Well, if there is hydrogen gas production, then the hydrogen gas is actually going to react with the iron that you have in the test tube. And if it reacts with it, what's going to happen is you're going to see a black color. Okay, there's going to be a black precipitate color that's going to form. So let me kind of show you, list some of the results and then show you the pictures for it. So let me just kind of draw right here, just real quick. So you guys remember we split the test tube into that top slant part. Okay, so this is the slant right here. Okay, that's an end right there. And then this is the deep. Okay, so basically the deep is going to help us figure out if glucose is fermented or not. Okay, the deep is for glucose. And then the slant part is going to be for lactose. Okay, slash sucrose. Okay, so let's take a look at the results together. So if the whole entire slant and deep part, by the way, the deep part, we actually call it the butt. Okay, so we refer to it, by the way, as the butt. So you're gonna see that term a whole lot. Okay, the butt, and so the butt portion is basically that bottom deep part. Okay. So if the slant and the butt, they're red pink, well, that means that none of the carbohydrates were fermented. This bacteria did not ferment glucose, and it was not able to ferment lactose or sucrose. It didn't ferment any of the sugars, okay? So if it doesn't ferment any of the sugars, the whole entire thing is going to be red, okay, or pink. Now, what if you get a pink top, okay, and yellow butt? Well, yellow means acidic. Remember how I said most bacteria, when they ferment glucose, they produce like an acidic, or, you know, they produce an acidic product. So, by the way, why is it changing color? Because there's another thing added, you guys, to this test tube, and that's the pH indicator. And so, as I said, the pH indicator usually changes color, you know, when your solution or whether your media basically turns acidic, basic, or neutral, it changes color. So we do have a pH indicator in here. And so if the butt changed color, and now it's yellow in color, that means that glucose was fermented. Remember how the butt is going to test for the glucose fermentation? So if the butt turns yellow, that means it's positive for glucose. But if the top is pink, remember what is the top part? Lactose and sucrose. So if it remains pink, well, that means that the bacteria is not capable of fermenting lactose or sucrose. Okay, so it just means that your bacteria is only capable of fermenting glucose. Now, what if the whole entire thing turns yellow? Now, if the whole entire thing turns yellow, well, that means that my bacteria is capable of fermenting glucose. Okay, and it also tells you that your bacteria is capable of fermenting lactose and sucrose, or maybe only lactose and maybe only sucrose from that top part. So this is how you would kind of like write it out. So you would say it's positive for glucose, right? It's positive for glucose fermentation. And, right, make sure that you say and. It's not or. Over here, it is very important, you guys, that you say and. Okay? So it's capable for fermenting glucose. Is that it? Nope. And it's also capable of fermenting lactose and or sucrose. Okay, guys? Now, the wording is super important. Okay? Because again, why here am I saying and slash or? Because I'm not sure, you know, this top part, you guys, has, you know, this top part is testing for two things: lactose and sucrose, or. Like, we don't really know when we get this yellow part up here, you guys, up at the top, we're not very sure whether both sugars are being fermented or just one of them. We have no idea. And, you know, from this test right here, we can't tell. How can you be sure? And you're like, okay, I want to make sure whether it's fermenting sucrose or whether it's fermenting lactose or whether it's fermenting both. How can I do that? Well, is there another test that you can do that tests one sugar at a time? Yes, the very first test we talked about, the carbohydrate fermentation test. Remember how we said with the carbohydrate fermentation test, we usually add only one sugar at a time? So you can do that, right? You can actually do the sugar carbohydrate fermentation test, you know, with the triple sugar iron test, okay? And that would help you determine, you know, specifically whether your bacteria is capable of fermenting sucrose and lactose, or, you know, only sucrose or only lactose, okay? But if you're doing the TSI test and the whole thing turns yellow, then you have to word it exactly like this. Okay, guys? So for you to get, you know, full credit on, let's say, like on a lab quiz, you have to word it exactly like so. Okay? Now, if, remember how I said the if there is hydrogen sulfide gas production, then it's going to react with the iron that we have in here. And if it does, then we're going to get a black color. And the black color is usually down at the bottom. So if you see black color down at the bottom, that means there is hydrogen gas being produced. Now, it's not a must, by the way, if you're it's not a must whether, like, if your bacteria is fermenting a certain sugar, hydrogen sulfide is going to be produced. No, sometimes it is produced and sometimes it's not produced. So let's kind of take a look at the results together. So right here, here's the slant part and here's the butt part. Now, if the whole entire thing stays pink, um, that means that none of the sugars are being fermented. None of them. Okay, so that means negative for all the sugars, okay, that are in here. And what are the sugars that are in here? There are three of them. There's glucose, lactose, and sucrose. So that means that none of these three sugars are being fermented. Now, notice how the bacteria is growing though. Okay, there is bacteria growth. Okay, so yes, you can have bacteria, okay, growing on this slanted part right here, but you know, it's not using any of the carbohydrates. Okay, it's using other nutrients in here, okay, for it to be able to grow. I mean, it must be using some other type of nutrients that are included in this test tube, okay, or in this medium, okay, but it's not using the carbohydrates. Now, what if you have, and again, remember how we always kind of look at it like this, okay? So the butt part is going to help us determine glucose fermentation. So if the butt part is yellow, well, that means that your bacteria is capable of fermenting glucose. Let's take a look at the top part. See how the top part is still pink? That means it's not fermenting lactose or sucrose. Okay, so right here, this test would only be positive for glucose fermentation. Okay, now let's take a look at this one. So again, here we have the butt and then we have the slant. Notice how the butt is yellow. So again, always the butt, think of glucose. Okay, so that bottom portion right here, if it's yellow, that means my bacteria is capable of fermenting glucose. Okay, let's take a look at the top part. See how the top part is also yellow? Well, that means the and, right, means your bacteria is also capable of fermenting lactose and or sucrose. Okay. Now, what if right here, again, let's do our division, okay? Notice how down at the bottom it's black. Okay, when you see a black color, what does that tell you? It tells you that your bacteria is capable of producing a gas, hydrogen sulfide gas. Let's take a look at the top. Notice how the top is pink. Well, that means that your bacteria did not ferment lactose or sucrose. Okay. Now, notice how the black color right here for the hydrogen sulfide is kind of taking over. So you can't really tell whether the glucose is being fermented or not, right? Because if glucose is being fermented, I mean, you're supposed to see the yellow color down here. But notice how the black color is kind of masking over. And so you can't really tell whether glucose is fermented or not. So we'll never give you you're never going to be given like a question on a quiz or so that's going to ask you, like, if you're given this image right here, I'm not going to ask you whether glucose is being fermented or not, right? Because you can't really tell. Okay, there's a high possibility that yes, glucose is is fermented, but again, we can't be 100% sure. So if you're given like a result like this, you just say that it's positive for hydrogen sulfide production. And then of course up here you would say it's negative for lactose and sucrose fermentation. So again, if you're given images of the TSI, you have to first of all identify that this is a TSI test. And then, of course, you're going to have to interpret the results and explain what they mean. And so for the slant test, as I said, we usually use a loop, right? And for the TSI, we use the loop and the needle. Okay, now there's another slant test, and this one we just use a needle to go ahead and inoculate. You're just gonna inoculate your bacteria on the slanted part right here. We don't use a needle with this one, just a loop, and this is called the citrate test. Okay, and this is how it would look like uninoculated. It's kind of like that green, it's kind of that greenish color. Okay. So we'll talk about what this test is used for. But there's another test called the urease test. And it kind of looks something like this. And again, you go ahead and you use your loop to inoculate the bacteria on this slanted part right there. Urease tests, they usually come in small test tubes. That's why the image right here is small, because the test tubes for the urease slants, they are usually pretty small in size. Okay, so just gonna keep that in mind. And again, this is how it would look like if it was uninoculated. Okay, so again, this is how they would look like uninoculated. And so now we can go ahead and start talking about the citrate and the urease test. So the citrate test, we want to figure out whether your bacteria is capable of using citrate. Okay, and citrate is used, by the bacteria as a source of carbon, okay, and as a source of energy. So if your bacteria is capable of using citrate, to get some energy and to use it to basically get its carbon source, then what's going to happen is it's going to turn blue in color. So if it stays green, by the way, then that means it's negative. Your bacteria is not capable of using citrate. But if it turns into that, blue color right here that you're seeing, that means that it is positive. That means that your bacteria is capable of using citrate. Okay. Now, the urease test, as I said, they come in those like tiny, kind of small test tubes. And what we're trying to figure out is whether your bacteria has the capability of producing an enzyme called urease. And what does this enzyme, what is this enzyme for? Well, when bacteria produces this enzyme called urease, the bacteria would have the ability to break down urea into ammonia. And ammonia is, like cleaning products have ammonia. Okay, so this is kind of the same ammonia that would be produced. And ammonia is basic. Um, it's basic in color. So usually in those slants right here, there's also a pH indicator that's added. So remember how, for example, with the phenol red, the pH indicator, when you get like a pink color, that would be a basic medium. Okay, so let's take a look at the results together. If you did the urease test and it stays yellow, that means that your bacteria is not capable of producing urease. Now, if your bacteria is capable of producing down urease, again, what is it going to do with urease? It's going to use that urease enzyme to break down urea into ammonia. Okay, and ammonia is basic, so the medium is going to turn pink. Okay, remember how pink was basic? Um, so therefore, again, because there's a pH indicator in there, when the pH of the solution, or sorry, like of the medium goes ahead and changes, that again, pH indicator is going to indicate that change that just happened. So um, if you get a pink color for the urease test, that would tell you that urea is breaking down into ammonia, and that's because the bacteria is producing that urease enzyme. Okay. Now, there are also tests done on plates. Okay, so let's take a look at some plates and how they look like uninoculated. So this is the EMB plate, and usually it's burgundy in color. Then we have another plate called McConkey, and it's usually kind of like that rose color. And then this is the TSA. We talked about TSA before. Remember the tryptic soy agar? Remember how we said the tryptic soy agar? It's an enriched medium, okay? Usually we use it as a control, okay? So let's talk about the EMB plate first. And let's talk about this term first before we discuss the EMB. Now, early on, early during the semester, we talked about how we have something called a standard medium, right? Kind of like a nutrient agar is a standard medium. We talked about a selective medium. What is a selective medium? We said that a selective medium, it's basically allows certain bacteria to grow and it inhibits another group of bacteria. And we kind of talked about Sabdex, right? And we said Sabdex is a selective medium because it selects for fungal growth, okay? Kind of inhibits bacteria growth. And then we also said that there is an enriched medium like the tryptic soy agar or the TSA. Now there is another, type of medium, and it's called the differential medium. And the differential medium is basically going to help us differentiate and distinguish by the appearance. So if you have a plate right here, this is an EMB plate, which is a differential plate. Notice how, based on the color right here of the colony growth that you're getting, you can actually differentiate between the bacteria. And that's the purpose of a differential media. It's going to help you identify the bacteria based on the appearance of its colonies. Okay. Or basically how, the surrounding media around it would look like. Okay. So one of the differential plates or the differential media, I should say, the differential media that we use is the EMB plate. And the EMB plate, first of all, comes in that burgundy color. Okay, see this background color right here? This is the uninoculated color, right? That burgundy color. Now, by the way, you can go ahead and divide this plate into several quadrants, and in each quadrant, you can go ahead and inoculate a different type of bacteria. Okay, now the EMB plate is selective. okay? Meaning that it's going to allow certain bacteria to grow on it, and it's going to inhibit certain bacteria to grow on it. What type of bacteria can grow on an EMB plate? Gram negatives. Okay, what does that mean? It means that gram positives cannot grow, cannot get cultured on an EMB plate. So if you see growth, notice how right here on this EMB plate, I have growth in that quadrant, growth, and here there's growth in that third quadrant. That means that all of these bacteria, they're all what? They're all gram negative, okay? Now, but notice how they look different, okay? So this tells us that the EMB is differential, okay? It is going to help us differentiate based on that color change that we are seeing. Now, what is it going to help us differentiate? It's going to help us differentiate those bacteria that are capable of fermenting lactose and those that are not, okay? And again, how are we going to be able to tell that? Based on the color change. So let's kind of interpret, the results. Now, if you get no growth, okay, if that burgundy plate, you look at it after you've incubated it for 48 hours and you see absolutely no growth, no lawn of bacteria, no colonies, just nothing. Well, what does that mean? It means that most likely, the sample that you inoculated your EMB plate with was what? Gram positive. And that's why nothing grew, because gram positives are inhibited from getting cultured on an EMB plate. Okay, so if you don't see growth, well, that means that it was a gram positive bacteria. Now, if you do see growth, what does that tell you? Right away, you know that it has to be gram negative. So any growth you see right here, you can see like there's growth on this plate, there's growth on that plate. Any growth you see, it must be gram negative. Okay, it's a must. It has to be gram negative. Okay, now how can I differentiate? I mean, okay, I know that those three are gram negatives. Okay, but how can I differentiate between them? Now, if the growth is pink, okay, guys, again, see how the plate is burgundy? So if you see this kind of pink color, okay, I mean, it's pretty obvious, right? If you see this pink color, that means that your bacteria is capable of fermenting lactose. Okay, so bacteria that are capable of fermenting lactose, they're going to turn the EMB plate from that burgundy color to kind of that pinkish color right here. Okay, now if you see green, see right here, that metallic green color, okay, what does that tell you? It tells you that your bacteria can ferment lactose, but they can ferment it pretty fast. So the rapid lactose fermenters. What's a type of bacteria that can ferment lactose? E. coli. Okay, so E. coli, that enteric bacteria, okay, is a rapid lactose fermenter. So notice how the EMB was helpful in allowing me to differentiate between my bacteria. So I look at this plate and I know any growth on an EMB plate must be gram negative. But how can I differentiate between them? Based on the color. So this is going to be a lactose fermenter bacteria. This one is a rapid lactose fermenter bacteria. Okay, well, what about this one? This one is, it's not pink, it's not green, but I mean, there is growth. So it is a gram negative bacteria, but it's not capable of fermenting lactose. So if a bacteria is not capable of fermenting lactose and it's growing on an EMB plate, then it's a gram negative, but it cannot ferment lactose. Because if it was capable of fermenting lactose, you would either see the pink color or you would see the metallic green. Okay, if you don't see either color, then it just tells you that it's a gram negative that doesn't have the capability of fermenting lactose. Okay. There is another test or another medium, that we use, and this is the McConkey. Okay, the McConkey plate. And the McConkey plate starts off like that rose colored plate. Now, the McConkey plate is selective. So again, it selects for gram-negative. So again, gram-negative bacteria can grow on an EMB plate, and they can grow on a McConkey plate. Okay? Gram-positives will not be able to grow on a McConkey plate, and it's not going to grow on an EMB. Now it's also differential also for lactose fermenters. So notice how you can actually do two tests that would tell you kind of same information. You can do the EMB test and you can do the what? And you can do the McConkey test. Okay. So the McConkey test is also going to help you differentiate lactose fermenters from those that cannot. But the colors are going to be different, right? So first of all, you start off with the McConkey plate, which is kind of that rose color. Okay, if you see growth, notice how right here I see growth on my McConkey plate, then I know right away it's gram-negative that's growing. Okay, now how can I tell whether it's fermenting lactose or not? So let's take a look at the results together. So first of all, if you don't see any growth, like nothing on your plate, then that means it's gram-positive. Okay, if you do see growth, then it means it's gram-negative. Now, if you see a pink color, so notice how again it starts out, it is starting out kind of like pink rosish, but notice how this part is even more pink. So if it's even more pink than what you started off with, then that means the bacteria is a lactose fermenter. Okay, so if it is a lactose fermenter, your McConkey plate is gonna turn pink. If it doesn't turn pink, okay guys, if you do see growth, okay, you do see growth, but you don't see the pink color, that means that it's a gram-negative, but it's not capable of fermenting lactose if you don't see the pink color. Okay guys, so that's the McConkey plate and the results for it. Now, let's talk about how do we use a dichotomy ski. Okay, so dichotomy ski looks something like this. It's kind of like a chart, and you basically take the results that you just received after doing all of the biochemical tests, and you basically try to figure out what bacteria you have. Okay, so we usually start up right here. The very first thing you want to know whether your bacteria is a bacillus under the microscope or whether it's a coccus. Okay, now if it's coccus, then you would go down right here. If it's bacilli, you go and start move your chart down here. Now remember, this whole PowerPoint we were talking about enteric bacteria. So we know three things about enteric bacteria. Remember, they are gram-negatives, they're bacilli, okay? And so therefore, by the way, if you look at the chart down here, okay, see how this is the chart for gram-negatives, okay? Because there's another chart for gram-positives, okay? But since again, right here we were talking about enteric bacteria, you were given right here this gram-negative chart, okay? So first thing, figure out the shape of the bacteria. Okay, so let's say that the shape of your bacteria under the microscope is a bacillus. So you would go to this side of the chart. Okay, next you would look at the first test, the oxidase test. Okay, and you would look at your results, right? So you would have done an oxidase test, okay, in the lab. And you see how right here it says negative or positive? That means that after you've done your oxidase test, did your oxidase test turn negative or did your oxidase test turn positive, right? So you would do the determination, okay, based on your result. Now, notice how there are several tests, by the way. So there's the oxidase test, there's the lactose, meaning whether your bacteria is capable of fermenting lactose or not. There's the Indo Test. There's the Methyl red, the Vogus Prosker, the citrate. Okay. Now the thing is though, after you've determined the shape of your bacteria, should you jump to determining the result for the lactose test right away? No, you must go in order. Notice how after determining the shape, what is the first thing you have to determine? Oxidase. Okay, you cannot jump to lactose if you haven't figured out what is the result for your lactose, for your oxidase test. Okay. Now, if my oxidase test is positive, I would go down right here. I would figure out, then whether my bacteria produced gas from glucose or not. If it produced gas, then this is my bacteria. If it didn't produce gas, then my bacteria is Vibrio. But if my oxidase test is negative, okay, then I would follow the arrow. Okay, so if my oxidase is negative, let's say my oxidase is negative, okay, which way am I going to go? I'm not going to go this way, okay, I'm going to go down this arrow, okay, so go down this arrow, and then now you should what? Interpret your lactose results, okay, so again, go in order. So shape, and then go down here to the oxidase test, figure out negative or positive. Okay, if it's negative, we're going to move down here. Then we're going to figure out the lactose. Okay, let's assume that your lactose is negative. Okay, follow the arrow. So if my lactose is negative, follow the arrow. Then what's next? I should look at my methyl, red, and Vogus Prosker tests. Okay. Based on your results, let's say my methyl red was negative and my Vogus Prosker was positive. Then that would mean your bacteria is Serratia. Okay. If not, if your methyl red was positive, Vogus Prosker is negative, then follow the arrow. And you're gonna continue and take a look at urease, etc. Okay, guys? So this is how the dichotomous key works. You keep following the chart. Okay, look at the next test. Look at your results. Figure out which arrow. Okay, figure out which side you should be following. Okay. And at the end, you will be able to identify the bacteria. Okay. Now, one thing to note, okay, guys, is that remember how, again, in this PowerPoint, we were talking about enteric bacteria. And we said there are 3 things to know about enteric bacteria. Number 1 is that they are gram negatives. Number 2 is they are bacilli under the microscope. Number 3 is that they are oxidase negative. Okay. And as I said, they are found in our guts. Okay. So they could either be part of your normal flora, or they could be pathogenic. Okay. Now, those that are actually, those enteric bacteria that are in our guts, that are actually part of our normal flora, it turns out that they are going to be positive for lactose fermentation. They are capable of fermenting lactose. So you would go down to the chart right here, okay? First of all, I know enteric bacteria are bacilli, but again, after you take a look at them under the microscope and determine their bacilli, you will go to your chart, okay? And then you will have to do the oxidase test, but we expect them to be negative, right? But again, still do the oxidase test. You must still do the oxidase test. Okay, so if it's negative, you're going to go down right here. If your lactose is positive, okay, then I'll go down here, and then I'm going to do the the methyl red, the Vogus Prosker citrate, etc. So if your, let me kind of grab my pen. So if your lactose is positive, okay, guys, you're going to go basically down right here. Okay, that would mean that this whole entire part, okay, so meaning that these bacteria right here, Klebsiella, Escherichia, Enterobacter, and Erwina, those are basically considered normal flora. Okay, so these are my normal flora. Okay, why? Because those are what? Positive for lactose fermentation. Now, if your bacteria, okay, so let me kind of erase. If your bacteria, let's say you did the lactose test and it's negative. So if it's negative, I'm going to go down this route. Okay, what am I going to do? Well, I'm going to do the methyl red test. I'm going to do the Vogus Prosker. I might do the urease. Okay, so I'm basically going to, you're going to go down this route. Okay, so that would mean that they are, these could definitely, those are basically the pathogenic ones. Why? Because those are the negative for lactose fermentation. So Serratia, okay guys, Proteus, Salmonella, Shigella, those are pathogenic enteric bacteria. Okay, so Serratia, Proteus, Salmonella, and Shigella, those are pathogenic because pathogenic bacteria, they are negative for lactose fermentation. Okay, make sure that you do know that. Okay, so pathogenic ones are the negative for lactose fermentation, and positive for lactose fermentation would be part of your normal flora. Okay. Now you're going to be given, a bunch of results. Make sure that you know how to read the results. Make sure that even if you are, see right here, you're not given the name of these tests. But make sure that you can still be able to identify what they are, right? Just based on how they look like, okay? So you're given, those are the results. You're going to take a look at each one of them, determine what those colors are trying to, to show you or tell you. And then after you figure out the result for each one of those, you're going to go back to the dichotomy skis. And the dichotomy ski is given to you on this PowerPoint, and you're going to try to figure out what is the bacteria. Okay. And even though we didn't go over the oxidase test, we know that all enteric bacteria, they are what? Like, since we're talking about the Enterobacteriaceae, Enterobacteriaceae specifically, right? The family Enterobacteriaceae that we were talking about, they are oxidase negative, okay? And so you have also the EMB plate to look at, and then also some other, unknowns to go ahead and take a look at to analyze. Okay, and unknown B as well. Okay.